On the quiz, most people did really well. I only gave out one 10 out of the class. I don't know, I don't remember who that is, but um, on that first question, I wasn't looking for all of the ways bacteria affect your physiology. There's a lot, but I was just trying to get you to identify the ones that have been talked about in the podcasts. So the obvious one is bacteria help you digest your food, but they also and liberate nutrients in the food you eat. And they talked about that in the podcast, but they also talked at length about the interaction with your nervous system, the vagus nerve, and releasing serotonin and stabilizing mood and lowering cortisol. Anything generally, if you said something like that, you got the bulk of the points. But they also had mentioned how bacteria stabilize or help your immune system develop. So to get like all the points, I was looking for people to mention all those things. The immune system bit was I only had a half a point to that. And then second question was how do you inoculate yourself with bacteria? That was so they talked about, yes, babies put stuff in their mouths and they live in a world, but the main way is through birth, the birth canal. As a baby is proceeding down the birth canal, they get inoculated by the mother's perineum as they come out. That's the first way that you get your bacterial flora. So it's like I was pretty leaning on that. If you like, a couple of you didn't listen or didn't pick up the idea, and you lost some points there. And then I pretty much got all the points on the third and fourth questions, unless you didn't like actually critique. Critique means like say more than just a grade. Like tell me what you thought, and whatever you thought, you got points as long as you told me. I didn't. As long as there was some sort of, you gave me some kind of perspective. So that was that. As far as the lab from last week, it's not going to be due on Friday because we're going to be finishing it up by fully combusting the apples either this week or we may do it next week. The apples have been dehydrated and I have them sorted and everything, but I'm not exactly sure how that lab's going to work out because part of the time we're going to be doing the fermented foods, bottling our first batch of kombucha for the second ferment, and then starting a new first ferment with different sugars. And then we're going to be taking the last readings on our sauerkraut, and if any of those sauerkrauts are good, we will sample those. There are certainly some that are not. And then we will be cleaning that up, and we'll try the sauerkraut again, and we're going to do it this time. I'll give you a very detailed instructions on how to do that so that we get more consistent results. I was interested in seeing what the experiment, sort of each person's experimental approach to sauerkraut was going to yield. But so that'll be Friday, and there will not be anything due this Friday in lab. But there will be some write-ups coming for the common grounds the following week. Fermented foods is an ongoing thing that you'll have to write up at the very end. So there will be some questions and some data analysis that you'll have to do for the common grounds apple thing. We generated a lot of data, and it's mostly been tabulated now. If you haven't entered your data that you collected into the class spreadsheet, you should do so this week, and so that all the data is there for everybody to plot it on their own and then submit it to me. Okay, so today I want to, it's come to my attention that not everyone in the class is actually super familiar with the fundamental background of chemistry that you may need to understand some of the words that I'm going to use. So some of you are very familiar with basic general chemistry from high school, but I don't want to assume that anybody is. So I'm going to devote one lecture today to covering basic chemistry, unless it is. I can take a vote on it. I don't need to put a lecture into this, but if people think it would be useful for me to just go through the basic chemistry terminology, the basic chemistry concepts that underlie this, who would find that useful? Okay, a couple people, 3, 4, 5, 6, like half the class, and then the others are. So for those of you who aren't super in need of this, I apologize, but I'll try to be brief. All right. So to begin with, there's this notion of the hierarchy of complexity. Life itself is organized into these levels, and here's an organism. That organism could just as well be a plant of some sort, or some other animal, or what have you. But we're human, so there's a human baby there. And this notion that complex structures are composed of simpler parts comes from our friend Aristotle, whom we all love. The other sort of seminal idea here is Democritus. It came from Democritus, who hailed from the same time period. And he believed that matter was made up of these things called atoms, which he called atoms, which are indivisible subunits. In fact, this idea that Democritus had was at odds with another of Aristotle's beliefs. Aristotle himself believed that matter was infinitely divisible, that you could take matter and break it down into infinitely smaller parts. They both believed in the power of thought experiment, but they had sort of conflicting results from those experiments. So the actual level of the actual ladder of life here starts with atoms, Democritus's atoms, and we'll understand what those are soon enough here. And they combine to form molecules. So those two levels are going to be the rungs on the ladder that we're going to explore today. And then on Wednesday, we'll move into macromolecules, proteins, and carbohydrates and lipids, and that macromolecule is actually DNA, which we will talk about in some detail. These macromolecules form organelles, which are then components of cells. Look at that. My diagram is off the slide, isn't it? So there we go. Let's get it back on there. Cells then combine to form tissues, and there are 4 tissue types. Each of those tissue types you'll see that in more detail later. But those tissue types combine to form the various organs that make up a plant or an animal. Organs can work as organ systems in organisms such as ourselves. And there I'm depicting the GI tract. And then all these organ systems come together to form the organism. All right, so let's focus on the first rungs of this ladder. Yeah, there's Aristotle. Don't we love him? So we probe these different levels of this hierarchy with these different techniques. So the unaided human eye that you see down here in the lower right-hand corner, I have a laser pointer, a dead laser pointer, can get down as low as a human oocyte. Oocyte being a human egg, the egg that is produced in the ovary of women. So a little bit larger than 100 micrometers, actually about 10 micrometers is about 10 micrometers is the size of a human egg, and you can see with sharp eyes, you can see it's the smallest thing that you can distinguish with the human eye. So then compound light microscopes, that yellow area, can get down to about maybe the largest virus. Maybe the largest virus can be seen with a compound light microscope. And then they have these two techniques, the transmission electron microscope and the scanning electron microscope, that we use to get very beautiful pictures of the very small things that our world is made of. Proteins, molecules, certainly viruses, DNA can all be probed with the transmission electron microscope. I have a pretty interesting story about the scanning electron microscope, but maybe you'll have to get it out of me sometime outside of class. I'm going to keep moving. All right. So, beginning the high school chemistry review. Atoms. An atom is made up of a nucleus, which has two types of subatomic particles in it, the proton and the neutron. They combine to form the massive core of an atom. The nucleus is surrounded by a cloud of electrons. So a proton its mass is one atomic mass unit, which is defined as 1/12 of the mass of one atom of carbon-12. This is not to say that an atom of, for example, helium which would have two protons and two neutrons in it, that's not going to equal exactly 4 atomic mass units for reasons that I'll talk about. But in general, a proton is about 1 atomic mass unit, and its charge is a +1. A neutron weighs the same as a proton. However, it is neutral, as the name would lead you to believe. Electrons are negatively charged. They have a charge of -1, so equal and opposite to a proton. However, they are mostly energy. Their mass is extremely minuscule, just 1/1800th of the mass of a proton. So when you're measuring the overall mass of an atom or a substance, the contribution to the mass from electrons, not electrons, is quite small. So I'm going to just fix that now because I will never remember it. All right. All right, so basically here, remember, this is a food class. Food is made up of atoms that have protons, neutrons, and electrons in them. An atom, we talked about the definition of an element already. So an individual atom is certainly going to be an element, something that is not in combination with any other atom. Here are 3 examples. So fluorine has 9 protons. Any atom of fluorine that you're going to run across, by definition, has to have 9 protons. If it does not have 9 protons, it is not fluorine. Likewise, cobalt, an atom of cobalt, another essential element to the human, has to have 27 protons for it to be cobalt. If it does not have 27 protons, it is not cobalt. And then again, likewise, iodine has 53 protons. If it does not have 53 protons, it is not iodine. So the number of neutrons does not have to equal the number of protons. In fact, it often does not. But it's in the same sort of range. They tend to be similar numbers. So fluorine, the most stable, what's called isotope of fluorine, has 10 neutrons. That means that its mass number is 9 from the protons and 10 from the neutrons. So its mass number is 19. The atomic number is 9, which is the number of protons, but it's also the number that it ranks on the periodic table, because the periodic table is put in order of increasing atomic number or increasing number of protons in the nucleus. The exact mass of this atom is actually not 19, though. It's 18.9984 atomic mass units. And this is because as you fuse the different numbers of protons and neutrons together into the nucleus, some of that mass gets converted to energy to hold that nucleus together. And so as you make a nucleus larger or smaller going down the periodic table, you're going to have slightly different energetic needs of construction. And so carbon-12 is 12 AMU by definition, but every other atom are just approximations that we're making based upon measurement, okay, and then comparison relative to carbon-12. You guys understand that? Follow that? You're going to need that to understand isotopes. We need isotopes to be able to understand a couple of the labs that are coming up about C3 and C4 plants. So this is one of the reasons I'm going through it. Am I losing anybody? Okay, good. So isotopes. Isotopes of an element all have the same number of protons. For example, chlorine. We have chlorine here. Chlorine has 17 protons. Always. That's what makes it chlorine. It has 17 protons. However, you can pack a different number of neutrons in there. Okay? We have a chlorine atom that has 18 neutrons. That's stable. And there's also a chlorine atom that has 20 neutrons. That's stable. Thus, although the atomic number for any atom of chlorine is going to be 17, the mass number for chlorine is 35 when it has 18 neutrons and 37 when it has 20 neutrons, because 17 plus 18 is 35 and 17 plus 20 is 37. So we see the exact masses of those next to that. Now, when you take a scoop of chlorine out of existence and you were to look at the relative numbers of chlorine 35 versus chlorine 37, 75%, roughly 75, 76% of the chlorine atoms would be chlorine 35, and about 24, 25% would be chlorine 37. All right, these are the relative isotopic abundances. All right, they're called the relative isotopic abundances. And chlorine, the relationship between chlorine 35 and chlorine 37 is they are isotopes of one another. The word isotope means isotopes of the same element have the same number of protons but different number of neutrons and mass number. All right. And we're going to see how that is pertinent down the road when we look at carbon isotope fractionation in C3 and C4 plants when comparing corn and rice. Yeah?
What's the mass on the periodic table?
Below, yeah. So, for example, there in chlorine, you see a number, it says 35.453. That's the average atomic mass. What they do is take so in this little table in the bottom, they would take the exact mass and multiply it by the percentage of its natural abundance and add all those up, and that would be the effective mass that you would see if you were looking at a sample of them. All right? Does that make sense?
Yeah.
So that's it. You could... Yeah, if you were to look at it, so look at that, 35.45, right? And you know that the isotopes are carbon 35, chlorine 35, and chlorine 37, but maybe we don't know the percents of each of those. And you see that it's 35.45. Well, that's a lot closer to 35 than 37. So you know that chlorine 35 must be the predominant isotope. All right. The number, and I'm not going to get too deep into some general chemistry for you, but that number is the average atomic mass, but it's also what's called the molar mass. And it has two meanings. Molar mass means how many grams would a sample of that stuff weigh if you had 6.022 times 10 to the 23rd atoms of that stuff, or a mole. That number is called a mole. But that's a bridge further than I need to take you guys. Okay? Yeah. Any other questions? Cool. So, take home here. Food is made up of atoms that have different isotopes. So here in this ridiculous picture of some cookies, I have an atom on the left that has two protons, must be helium, and it's got two neutrons, protons in red, neutrons in blue. And then the other side I have two protons, so it's the same element, but it's a different isotope because there are three neutrons in it, three blue dots. Okay? So one would be like helium-4, and the other would be like helium-5. I don't even know if those exist. These are cookies. Okay? Basic. Great. So I had kind of covered this one day in lecture, but this is how to interpret chemical formulas. So water, H2O, that chemical formula, that means it has two atoms of hydrogen and one atom of oxygen. Sodium carbonate is going to have one atom, so it's NaHCO3. That means there's one atom of sodium, one atom of hydrogen, a carbon, and three oxygens that are all bonded together, and they can either be covalently or ionically bonded. We'll see what that means in a moment. But so we've stepped up from atoms up to molecules, up this ladder of life. All right. So the way in which all these atoms can combine are dictated by, or perhaps described, I guess, by where they are on the periodic table. And this periodic law, Mendeleev, Dmitry Mendeleev, a Russian chemist from the 19th century, his periodic law that as you go up in atomic number, there are repeating chemical traits that have a sort of periodicity to them. All right. So both lithium and, for example, lithium and sodium are in the same column, the same group in the periodic table, and they exhibit similar chemical properties, similar bonding properties. So lithium and sodium both combine with phosphorus in a 3-to-1 ratio, or sulfur in a 2-to-1 ratio, or with chlorine in a 1-to-1 ratio. And they show similar chemical behavior because of that. All right. This is all because of what is called valence electrons. We'll get to what valence electrons are now. All right. So we talked about the nucleus of atoms and how that gives us what isotopes are. Let's talk about electrons, because electrons are really where the action is. Electrons are where the action is. Electrons are going to dictate the kind of chemical reactivity that a particular atom is going to have. Here is an atom of argon, and so the electrons that are arrayed around the nucleus of argon arrange themselves in shells. Arrange themselves in these what are called shells. The first shell, and I should say this, each shell is itself subdivided into what are called orbitals. All right. So shell number 1, the first shell, has only one orbital in it. Okay, and every orbital can only fit 2 electrons. So shell number 1 can only fit 2 electrons. Shell 2 has 4 orbitals. Each orbital can contain 2 electrons, but there are 2 types of orbitals. It gets confusing. We don't need to get into the details of that. There are 2 s and then there are 2 p orbitals in shell 2. 2s can only fit 2, and then there are 3 2p, so that can fit 6 in the p orbitals in shell 2. That's more detailed than I actually wanted to go into. Okay, so the point of this slide is trying to introduce you to the notion of the octet rule. So for most of the atoms that are going to concern us, that is atoms that make up the human body, there's the smaller atoms towards the top of the periodic table. They will follow this octet rule. The heavier atoms don't. Once you get down to the larger shells, they don't fit the octet rule anymore. But the octet rule is basically that atoms want to have a complete outer shell of electrons. All right, and the octet rule says that complete shell is 8. All right, we want 8 electrons in that outer shell. So as we move across the periodic table, lithium is only going to have 1 electron in that outer shell. Beryllium is going to have 2. Boron will have 3, carbon 4, nitrogen 5, oxygen 6, fluorine 7, and then neon 8. Neon is one of the noble gases. And noble gases are called noble because they are aloof and do not interact with other atoms. Why? Because they have a complete outer shell of electrons. It's the electrons in this outer shell that are going to interact with other atoms to form bonds. All right? So the bottom line here is food is made up of atoms that have electron shells. And those outer electron shells are what are going to be doing the chemistry, going to be doing the bonding. You want to get to 8 electrons in those outer shells. Yeah. Okay. Is that clear? You guys follow that? All right. So let's look at fluorine, for example. Fluorine it has 7 valence electrons, and we find it as F2 out in nature. When you find fluorine gas, it's F2. The way this works is one fluorine atom, which has 7, the other fluorine atom has 7. They come together and they each share one of their electrons with the other and fill a joint orbital. So there's these 3 orbitals around each that have the full complement of electrons in them, but there's one orbital that's not fully complete yet. It doesn't have all its electrons. They come together and they each donate 1 electron into a combined orbital. All right? That is what we call a covalent bond. All right? So when 2 atoms dump an electron each into a shared orbital, completing that orbital, that is a bond that's holding them together. And they call it covalent, meaning co together, valent. The valent electrons are the ones on the outside, the outermost shell electrons. So their valence electrons are being shared together. They're covalent.
What's the vessel that the electrons are shared in?
Say that again?
When they share, but not share, but
Yeah, they are sharing. Yeah.
Yeah, trade those 2 electrons in the middle. Would you say that spaces?
Yeah, that is a shared orbital. Yeah. I'm trying to fit kind of a whole semester of general chemistry into a lecture here. So I'm purposely not going into a lot of the details about exactly what orbitals look like. But they're sharing electrons together, a pair of electrons. Each pair of electrons that gets shared is 1 bond. So if you have 2 pairs of electrons, meaning 4 electrons, that's a double bond, right? Or if you have 3 pairs of electrons that get shared, that's a triple bond. Fluorine here has a single bond. And so now each of those fluorines, if you look at the sort of Venn diagram of electrons, each fluorine has 8 electrons around it. It's really happy. It's satisfying the octet rule. And each atom has the electronic structure of neon here. If you look at neon in the corner, neon has 8 electrons around it. It's a noble gas. It's on the far right-hand side of the periodic table. Its valence electrons are fully symmetric, totally occupied, very happy. And that's what fluorine is attempting here. So let's look at another example of this. Water, H2O. So oxygen has 6 electrons in its outer shell. 2 electrons, so oxygen's atomic number is 8. 2 electrons are in its shell number 1, and shell number 1 is full and done. And then the other 6 electrons fill up shell number 2. So its valence electrons are in shell 2. There's 6 of them. But it wants 8. Well, there are lots of protons out there, lots of hydrogens that are willing to share their single electrons with the water. And in just the same way as 2 fluorine atoms shared their electrons into a single orbital to form a single bond covalently, each of the hydrogen atoms donates an electron into orbitals, the 2 unpaired orbitals in the oxygen. So now if you look at the oxygen on the right-hand side has 8 electrons arranged around it. It satisfies the octet rule. The hydrogen doesn't need to have 8 because its valence electrons are in that shell 1, which only fits 2 electrons, but it's satisfying its own completed electron shell, right? So it looks like electronically it looks like helium now, although it's a hydrogen. So if we look more closely on the bottom, 2 of the electrons on the right and 2 on the left in red are bonding electrons that are covalently being shared between oxygen and each of the hydrogens, holding the hydrogens in place. And then there are 2 non-bonding pairs of electrons, just lone pairs of electrons that are sticking off the oxygen, and they act as electronegative centers, so negative charge centers on the butt end of the oxygen. That's going to become important when we talk about hydrogen bonding and what that means. But all right. Please louder. Okay, so any orbital can only have 2 electrons in it. A shell, which is made up of multiple orbitals, in this case 4, a shell can have up to 8 in the limited world that we're talking about, the top of the periodic table. For 2 electrons to be shared between 2 atoms, that is a single covalent bond. If 2 atoms are going to share 2 sets of electrons, that's a double bond. You follow me? Yeah, okay. Everyone else? All right. So, in general, a chemical bond is just when 2 atoms are attracted enough to one another to stay together. It doesn't have to be covalent. Covalent is one way that 2 atoms can be attracted enough. They're sharing electrons. It's kind of like the Brady Bunch. The Brady Bunch was like that, right? You guys ever watch reruns of the Brady Bunch? There was a family, a lovely lady, trying to raise kids, whatever. They both brought a bunch of kids to the family. They shared kids, and they both divorcees, shared kids, held them together. That's kind of like a covalent bond. There are different types of covalent bonds. We can have polar or nonpolar covalent bonds. And this is important because this is going to have profound impacts on the way that DNA works and proteins work, etc. And it's also going to have a lot of impacts on how food gets processed and what happens to food when we cook it. So we'll get to that. Polar covalent bonds. Well, I guess first, nonpolar covalent bonds are ones like fluorine, when the two partners, the two atomic partners who are each sharing those electrons, are pulling on those electrons with the same strength. So one side is not stronger than the other. All right. However, a polar covalent bond is when one of the partners in that bond that are sharing the electrons is pulling on the electrons harder than the other side. All right. So if we look at HF gas, hydrogen fluoride gas, very strong acid when it's in solution. That's what they use to etch glass. The stuff eats through glass. It's extremely polarized because fluorine really likes to pull electrons towards it. It really likes to pull electrons towards it because it really wants to satisfy the octet rule. It really strongly wants to get 8 electrons right around it. So it's very greedy, pulls the electrons towards it. We call that electronegative. It's very electronegative. It likes to be slightly negatively charged. It's happy when it's an ion. We'll talk about what that means in a moment. So, and then hydrogen, it's okay living without an electron at all. So it tends to get the short end of the stick in terms of electrons in an HF bond. So the hydrogen end of the atom is a little bit positively charged. So we have this dipole setup. So a little bit of positive over here, a little bit of negative over there. And that means it's polarized. So when you have a whole space, a whole room of these atoms bopping around, one hydrogen fluoride atom, the hydrogen on one can kind of get attracted to the fluorine on another, and they can kind of stick to one another a little bit. You guys see that? How it's sort of like a whole bunch of, it's not magnetic, it's electric, but it's kind of like a whole bunch of magnets that could be attracted to one another end to end. Okay? Any questions there? All right, so what do you need to remember? Food is made up from atoms that are bonded together by either sharing or donating electrons. And we'll see what donation means here in just a bit. So, if an atom is towards the left-hand side of the periodic table, meaning it has fewer than 4 valence electrons, then it can satisfy the octet rule by unloading those electrons. It's easier for it to unload electrons. Say it has, like sodium, maybe it only has 1. It's a lot easier for it to get rid of 1 electron than it is for it to pick up 7 new ones somehow. So this is different than covalency. Here on the left-hand side, we have sodium atom. Its first shell has 2 electrons. Second shell has 8 electrons in it. Third shell has only 1 electron in it. And this is a neutral sodium atom. It's neutral. There's not a charge. It's not carrying a charge, but that shell is kind of unstable. There's this electron that's just out there by itself, spinning around. It's not a very stable arrangement. It's not a very symmetric arrangement for the atom. So that atom is pretty happy to get rid of that electron. Give it to somebody else that's willing to take it. All right? And when that happens, you can get sodium where shell 2 is now the valence shell, not shell 3. Shell 2 is, and it has 8 electrons in it. Totally stable, totally happy. No problem in the valence electrons. So sodium ion, Na+, is quite a stable species. The sodium ion is a very stable species. But you can't just like throw an electron out the window, you have to have somebody pick it up, right? It has to go somewhere. You don't just have free electrons floating about. Let's look at another example. What about chlorine? Here's a chlorine atom on the left. It has 2 electrons in shell 1, 8 and 2, and 7 and 3. And it just needs 1 more to complete that outer shell. So chlorine is really happy to pick up an electron. And when that happens, chlorine becomes negatively charged, and but its octet rule is satisfied in that outer shell. All right, so its total charge is -1. Well, sodium metal and chlorine gas can combine really well, quite happily, to form sodium chloride, where sodium is lost an electron and it's positively charged. Chlorine has gained an electron, it's negatively charged. And because they're oppositely charged with formal charges on them, they like each other. They are attracted to one another. Opposites attract in the chemical world. All right. Guys, follow that? I know this is a whirlwind for some of you and probably a little boring for others. I apologize. All right. So that's what an ionic bond is. Here, so sodium unloads this electron to chlorine, becomes an Na plus and a Cl minus. They like each other because they have opposite charges, and they come together forming an ionic compound. There are no shared electrons. Chlorine has taken the electron from sodium. Sodium does not have it. Sodium is happy to get rid of it. They have negative charges and positive charges, which are balanced with one another. So when you form a salt, this is essentially a salt, a metal and some sort of counteranion, you get these crystals. And so here on the left is a crystal of salt that has been zoomed in. And what is that? So it's alternating sodium and chloride atoms, ions. Sodium is a cation and chlorine is an anion, like cathode and anode, positive and negative. Chloride is the cation sodium is the cation and chlorine is the anion. And they just alternate in this enormous crystalline grid. All right, this is what a salt crystal is. All right, so there's more to it than that, but remember, food has salt on it. So that's all the chemistry. I thank you for being patient as I whipped through that. Are there any people that have questions about that who didn't sort of understand what I was talking about there? That was background information. I didn't want to assume that you knew. This is a 100 level class, but you do need to know that to understand some of the biochemistry here. Okay. Well, great. That's the perfect background then to talk about this. It's going to be a little more interesting, perhaps. I don't know. General chemistry is interesting. The biomolecules of life and food. So there are basically 4 categories of biomolecules: proteins, carbohydrates, lipids, and nucleic acids. So we're taking another step up this chain here. And first we're going to talk about proteins. And proteins are important because they make up everything around you. The vast majority of the dry matter in your body is made up of proteins. And then we'll follow it up by carbohydrates next week. The rest of the week will be taken up talking about proteins. Next week will be carbohydrates and lipids, and then I'll talk about nucleic acids a little bit, and then we'll be on to that'll be the midterm. We'll be at the midterm. So these are the proteins, carbohydrates, lipids. Those are also considered to be macronutrients when you're talking about it in nutritional terms. They're the macronutrients. And then after the fall break, we'll pick up with micronutrients, coenzymes, vitamins, minerals, and all that. But for now, number one, proteins. All right, so just to review the nitrogen cycle, starting on the left, there's nitrogen gas that's floating around in the atmosphere, N2 gas, and bacteria in our environment are able to fix that nitrogen into NH4. Lightning does the same thing. Haber-Bosch can do the same thing. We talked about the different ways of fixing nitrogen, getting it in the soil. So ammonia can go back and forth between nitrite and nitrate, those NO2 and NO3 species down there. Ammonia is then converted by plants and bacteria into amino acids. All right, heterotrophs or autotrophs, pardon me, are able to take that ammonia and create amino acids, and like the purines and pyrimidines from the nucleic acids, other nitrogen-containing compounds are produced by plants and bacteria. And then animals take those amino acids and they can reduce them and give off ammonia or ammonia-related compounds. Or they can go through the metabolic processes of anabolism and catabolism. So amino acids are, so you've probably heard of the word anabolic before in the context of anabolic steroid. What is an anabolic steroid? Has anybody heard that term before? What does it mean? Does anyone want to venture a guess? What's an anabolic steroid? Yeah. Muscle growth. That's right. It's a steroid that helps the cell take amino acids and build them into protein. So that's the process called anabolism. Anything that is taking simpler subunits, biomolecular subunits, and then building them into larger, more complex structures, like amino acids into proteins, that is anabolism. The reverse process of taking proteins or anything else that's large, like a polysaccharide, for example, like glycogen, and breaking it down into the smaller subunits, that's called catabolism. So there are anabolic and catabolic steroids. Muscle builders certainly are more interested in anabolic steroids. But that's the process, anabolism and catabolism, going from simple amino acids up to proteins and back. All right, so that's just a review of the nitrogen cycle, letting you know how nitrogen is playing in, is the foundation for our discussion of amino acids and proteins. We'll pick up there next time. There are 2 podcasts on this week that I've posted. Please take a listen to those. They are very different and from one another. One is about kombucha, the other, which is what we're going to be doing on Friday, and the other is about proteins, which is what we're going to be talking about in the next couple days. So listen to those. Keep an open mind as you listen to them. I have my motives. Are there questions?